So now I'm going to go over the 20 different amino acids. So they're split into different groups. The first group I'm going to go over is the nonpolar and non-aromatic amino acids. So nonpolar, they're usually made of the carbon and hydrogens because they're aliphatic chains, and they're also non-aromatic because they're not conjugated. So the first amino acid is the most basic one, glycine. Each amino acid has a 3-letter code and then also a 1-letter abbreviation. You're gonna have to know these for the MCAT. So for glycine, the 3-letter code is GLY, and then the 1-letter code is G. So on the right, as you can see, each amino acid has the same structure that's in black. But then this green circle right here, that is the R group. So that's the group that will change between the different amino acids. So for glycine, it's the most basic one. It has a hydrogen. And then for alanine, the 3-letter abbreviation is ALA, and then A is the 1-letter abbreviation. And this has a methyl group as the R group. So as I'm going through these amino acids, the nonpolar chains, they're going to get a little bit bigger. So valine has 2 more carbons, valine VAL, and then V as the abbreviation has 3 carbons. You're going to have to know the basic structures of the amino acids and the abbreviations as well for the MCAT. And then leucine is the next one, LEU, and L as the 1-letter abbreviation. It has 1 more carbon attached to it. And these are all nonpolar, non-aromatic ones. And then finally, isoleucine, ILE, and then I as the isoleucine. So don't confuse this capital I for an L. All the 1-letter abbreviations, they're capitalized. So that this is an I, not a lowercase l. That has 1 more carbon. And then methionine, MET, this is usually the first amino acid incorporated into a protein. The 1-letter abbreviation is M, and this is a structure for it. It has a sulfur in the carbon chain. And then the last nonpolar, non-aromatic amino acid is proline, PRO, P, and you can always identify proline because of this 5-carbon ring in the structure with the amino group. So it forms a intramolecular ring, and because of the ring, proline, it's usually at the end of a chain or in the turns and loops of the alpha helix. So those are all the nonpolar and non-aromatic amino acids. Now I'm going to go over the nonpolar and aromatic amino acids. So these amino acids, they have a conjugated system in the R group. So the first one is tryptophan, TRP, and this is the first weird one. The 1-letter symbol is W. And you can identify it by these 2 conjugated rings. And then phenylalanine, PHE, and this is also weird too, it's F. You can remember it because PH makes an F sound. And this has a phenyl group attached to it. And lastly, this is a polar and aromatic amino acid. So it's polar because of this hydroxyl group at the end, this OH. It causes the amino acid to be polar because it's not just carbons and hydrogens in the chain. This OH hydroxyl group adds polarity to it. So tyrosine, TYR. It's also weird. The 1-letter symbol is a Y. And then, yeah, so it's polar due to the OH on the benzyl group. And then these amino acids, they're polar but non-aromatic, so they don't have the conjugated ring system. So the first one is serine, S-E-R-S, and it just has the carbon attached to the hydroxyl groups. And the next one, threonine, T-H-R-T. This has just an extra carbon attached to the R group compared to serine. And then these are also polar but non-aromatic, but instead of hydroxyl groups, they're amide groups. So first one is asparagine, ASN. This is also weird too. N is the single letter abbreviation, and it's an amide group as the side chain. And then glutamine, it's similar except it has one more carbon in the chain. Glutamine, GLN, and it's also weird. It's Q. So I usually remember glycine as G, and then this Q, it kind of looks like a G. So try to remember glutamine as the one that has Q because the Q kind of looks like a G. So later on, I'm going to go over how the amino acids either lose or gain H pluses due to pH changes, but I'm just going to go over it really quick over here. So these amide nitrogens in the side chains, they don't lose or gain protons due to pH changes, but the amino group in the actual amino acid, these NH3 pluses at the top, they do pronate or depronate depending on the pH change. And finally, the last polar but not non-aromatic amino acid is cysteine. Cysteine, CYS. C, it's different than the other amino acids because it has this thiol, and thiol or SH, the bond is weaker than a a bond formed by a hydroxy group, since sulfur is a larger molecule than oxygen, so it's less electronegative. So the bonds it forms due to the SH is weaker than if you were to form like a peptide bond with an OH to amino group. But cysteine is interesting because it can form disulfide bridges. So when you have two cysteines close to each other, it can form cysteine, which is the result of the disulfide bridge. So at the bottom, you can see two cysteines combined together and the two sulfurs bond to each other. But you can break this up using a denaturant that oxidizes this bond and reduces it, and oxidizes it in order to break apart the disulfide bridge. So cysteines, the disulfide bridges, they contribute to the tertiary structure of proteins. And then, so acidic amino acids, these are one group of amino acids that that the R group can pronate or depronate based on pH changes. So when the side chain is deprotonated, it can have a negative charge. So the first one is aspartic acid, ASP, and the one letter abbreviation is also weird. They're running out of letters, so they use D. And so this on the right is the deprotonated form. As you can see, the O minus in the green circle. So that carboxylic acid was deprotonated, so the O has a negative charge. And when it's deprotonated, it's called aspartate. So on the MCAT, they'll either use aspartic acid for the protonated form or aspartate for the deprotonated form. And I'm going to go over this more later, but just want to let you know that the pKa of this side chain of the aspartic acid is 3.90. And then glutamic acid looks similar to it. It just has an extra carbon in the chain. So the abbreviation is GLU or E. So just remember that D and E, the two letters close to each other, they're both acidic acids, acidic amino acids. And similarly, it's called glutamate when it's deprotonated and glutamic acid when it's protonated. So on the right is the deprotonated form with the O minus. And the pK is similar. It's a little bit higher than aspartic acid. It's 4.07 for the R group. And finally, the last group of amino acids, the basic amino acids. So these are the amino acids with side chains that when protonated, it can have a positive charge. So the first one is lysine, LYS, and the letters K. I try to remember as K is the letter after L, lysine. So leucine is L, but lysine is K. And the pKa of the side chain is 10.54. And then arginine, it looks different than lysine. So it has these 3 nitrogens in the side chain. So the 3 nitrogens, it's able to delocalize that positive charge once the side chain is protonated. The positive charge can be delocalized between those 3 nitrogens. So if you move some bonds around, you can move the positive charge to any of the 3 nitrogens in that side chain. And the pK for that side chain is 12.48, so it's pretty high. And arginine is ARG or also R. So arginine kind of sounds like R, the letter R. That's how I kind of remember that. Okay. And then the last amino acid is histidine, HIS, H. So it has this ring called the imidazole ring. The positive charge when protonated is delocalized between those 2 nitrogens in the ring. And the pK for that side chain is 6.04.